In this video, we are going to have some multiple choice objective questions under enzymes. Don't forget to like and subscribe after watching the video so you can receive every update from this channel. So let's begin. The biocatalysts which increase the chemical reaction are called. So which of this is a biocatalyst which increase the rate of a chemical reaction or a biochemical reaction? The answer is enzymes. Enzymes are basically protein molecules which act as catalysts to speed up biochemical reactions which occurs in the cells of a living system. Enzymes increase the chemical reactions by lowering the. So what effect does enzyme have on a chemical reaction? So they lower the activation energy of a chemical reaction. And the activation energy is the minimum amount of energy which must be overcome in a reaction so you can get a product from the substrate or the reactant. So the answer is B. Mostly enzymes are composed of. So in most cases, enzymes are made of proteins, or enzymes are proteins because they are made of amino acid sequences. Few enzymes are made of nucleotides, and they form class of or group of enzymes. You call them ribozymes. But most enzymes are made of amino acid, which makes them composed of proteins. The term enzyme was proposed by. So which of these was the first scientist that proposed the term enzyme? Well, a German scientist by the name Kuhn, he was the first scientist who discovered or who proposed the name enzyme after his fermentation research work. So the name enzyme was given to biological catalyst by a scientist called Kuhn. The molecules of nucleic acids act as. So in a chemical or in an enzymatic reaction, what does nucleic acids act? So nucleic acids act as coenzymes. Coenzymes formed from nucleic acids are the NADH, which is the FADH2 and FMN. So flavin mononucleotides, nicotinamide adenine dinucleotides, or flavin adenine dinucleotides are all nucleic acids that act as coenzymes that activate enzyme or protein enzymes in a chemical reaction. The site of enzymes where substrate is attached is called. So on the enzyme where a substrate binds is called an active site. So the active site is the site on the enzyme where the substrate binds and gets converted to the product. A lock-key theory about action of enzyme is proposed by. So which of these scientists proposed a lock and key model about the action of enzyme? This model says that enzymes basically have an indentation that is complementary in shape to the substrate. So which scientist proposed the lock and key model? It was the Fischer who proposed the lock and key model of an enzymatic reaction. The inactive site of enzyme is called. So the active site is the site that the substrate binds. Any other site on the enzyme where the substrate does not bind is called an inactive site. And those sites are of regulation and they are called allosteric sites. So inactive site of enzymes are called allosteric sites and they are meant for allosterism, which is the regulation of an enzyme activity in the course of the enzymatic process. The induced fate model theory about the action of enzyme was proposed by a scientist called Kochland. So Kochland proposed the induced fate model. This model says that enzymes basically have a site around it that binds to a substrate and induce a conformational changes on the active site so that that binds a molecule that induces a conformational change on the active site so that the substrate can bind. So the substrate, or let's say the molecule that binds the enzyme, induces a conformational change to the enzyme's active site. So it becomes complementary to the substrate where it can bind and get converted to the product. So this theory was proposed by a scientist called Kochland. The enzymes which consist of only proteins are called. So if enzymes are consisted of only proteins without any other conjugate, those are what we call the simple enzyme, or also called the apoenzyme. So if enzymes are made only of proteins, they are called simple enzymes or enzymes. If they get bound to a molecule, we call them a conjugated enzyme. If that molecule is not removable, we call that group a prosthetic group. So simple enzymes are basically enzymes which consist of only proteins. Enzyme molecules are, so which of these is or are true about enzyme molecule? It is heat sensitive but not pH sensitive. No. Enzymes are protein, so they undergo denaturation, and change in pH also affect its activity. So enzymes are sensitive to both pH and heat. So A is wrong. pH sensitive but not heat sensitive, it is wrong. Both pH and heat sensitive, it is correct. Neither pH nor heat, no. Chemically changed, no. So enzyme does not change in a reaction, but they just help to convert or speed up the chemical reaction by lowering the activation energy of the reaction. So enzyme molecules are both pH and heat sensitive. So with the Fischer, as we said earlier in the previous question, he proposed the theory of the lock and key model, which says that the active site of the enzyme is complementary in shape to the substrate. The Quotient, as we said earlier, proposed the induced fit model, which says that an enzyme binds a molecule which induces a conformational change to the active site that becomes complementary to the substrate so that they can bind strongly. So the induced fit theory or model was proposed by Quotient in the year 1959. Enzymes can react with acidic substances only as their proteins. No. Enzymes can react with alkaline substances only as their proteins. No. Enzymes react with acidic and alkaline substances both. So here the answer is C, because enzymes are made of amino acids which are made of both alkaline and acidic amino acids. So the alkaline ones react with acidic substrate, then the acidic ones react with alkaline substrate. So enzymes can react with both acidic and alkaline substances both. So regarding the nature of enzymes, which one of the following is correct? So the nature of enzymes, which of this is correct about the nature of enzymes? Enzymes are modified lipids. No. Simple enzymes consist of protein and non-protein part. No. Conjugated enzyme consists of non-protein part. No. Amylase is an enzyme which entirely consists of proteins. Yes. Amylase is an enzyme which entirely consists of proteins. So amylase is an enzyme which consists mainly of proteins. It has no non-protein part, or let's say it has no cofactor to activate it. But amylase needs a cofactor for its activation. So the answer for this reaction is D. In case of conjugated enzymes, the protein part is called. So for conjugated enzyme, as I said earlier, the protein part of the enzyme alone is called the simple enzyme or the Apple enzyme. In the case of conjugated enzymes, the non-protein part is called the prosthetic group. So if an enzyme is conjugated, the non-protein part, if it is not removable, then we call it a prosthetic group. But if it is removable, we can call it a coenzyme. If it's an organic molecule, like an essential ion, if it is an ion or an elemental ion, or let's say a cofactor for all. So the non-removable protein part, non-protein parts of enzymes are called a prosthetic group. Enzymes are classified into 6 different classes. So enzymes have 6 different classes. You can have oxoreductases, you can have transferases, you can have hydrolases, you can have isomerases, you can have ligases and lyses. So these are the 6 main broad categories of enzymes. Enzymes function to increase the rate of a metabolic reaction by, so this one, how does enzyme help to increase the rate of a reaction? They denature the substrate? No. They add energy to the reaction? No. They decrease the energy of activation? They increase the concentration of the reactant. So here they decrease the energy of activation, or they decrease the activation energy of the reaction, which is the minimum amount of energy required for the substrate to be converted to the product. An enzyme speed up a chemical reaction by, so this one they are similar. They regulating pH, act as a buffer, they prevent denaturation, or they lower the activity. So the answer is D for this one. Enzymes speed up the rate of a chemical reaction by lowering the activation energy of the reaction. The role of an enzyme in a chemical reaction is to, so what role does enzyme play in a chemical reaction? Do they emulsify fat? No. Do they prevent denaturation? No. They speed up a chemical reaction. So enzymes speed up the rate of a chemical reaction by, as we said earlier, by just lowering the activation energy of that reaction. Enzymes often they absorb fatty acids, they help in hydrolysis reaction, they serve as long-term source of energy, or they serve as structural framework of cells. No. So enzymes help in hydrolysis reaction. Example, if you have a maltose, you want to convert it, break it down to 2 glucose molecule, you need an enzyme like maltase to hydrolyze it to break the 2 or the disaccharide into the 2 monosaccharide. So enzymes help in hydrolysis reaction. They add water to break the bond. In the cell, enzymes act as, so in a cell, enzymes act as buffers, they act as catalysts, yes, they act as neurotransmitters, no, emulsifying agent, no. So enzymes act as catalysts in cells. Enzymes consist of chains of, so enzymes, as we said earlier, they are mainly proteins. So they are made of chains of amino acids. But few enzymes are made of nucleotides, and they are called the ribozymes. Compounds that are needed for enzyme to function properly are, so which of these is the compound that enzymes need to function properly? Is it buffers? Is it steroids? Is it vitamins or heavy metals? The answer here is the vitamins. But the vitamins are what is used in the formation of the cofactors that activate enzymes. The NADH, the FADH2, the FMN are all derivatives of vitamins. So vitamins help to function or to manufacture these cofactors in the body so that they can be used to activate enzymes. A non-protein molecule that aids the action of an enzyme to which it is loosely bound is called, so a non-protein molecule that is the action of an enzyme to which it is loosely bound is it an initiator? Is it a coenzyme? Is it a competitive inhibitor? Or is an enzyme substrate complex? So which of this is a non-protein molecule that aids the action of an enzyme to which it is loosely bound? So the non-protein molecule that aids the action of an enzyme to which it is loosely bound is called the coenzyme. So the answer is B. The area of an enzyme into which the substrate fits is called the active site, as we said earlier. The molecule that fits into the enzyme active site is the substrate. The substrate fits the active site of the enzyme. The active site of an enzyme is formed by a substrate, altered by heavy metals, altered by substrate concentration, or they are destroyed during its reaction with a substrate. So the active site of the enzyme can be altered by the heavy metals, because heavy metals they tend to interact with amino acids and changes the amino acid constituent of the active site, which alter it. So the active site of an enzyme can be altered by heavy metals. So let's look at the energy profile diagram here and see how best we can label it. If this is a diagram for an enzymatic reaction, that shows how energy varies as the enzyme reaction goes on. This is the beginning of the reaction. It gets to a transition state where the molecules here are called activated complex. Then we get the product. The difference between the energies of the transition state and the substrate is called the activation energy. So here, the first one is the normal profile for the uncatalyzed reaction, and the second curve is that of the enzyme catalyzed reaction. So you can see that the A is the substrate, the D is the product. You can see the B is the activation energy of the uncatalyzed reaction. The uncatalyzed is the reaction without the catalyst, without the enzyme. But when you add the enzymes, it lowers the activation energy. So here the C is the activation energy of the catalyzed reaction. Thanks for watching. Don't forget to subscribe so you can receive every update from this channel. Thank you.